Now we will discuss the fourth biomacromolecule, that is our nucleic acids. See, I'm not going to discuss the third biomacromolecule, that is lipid, because we have already discussed in the classroom. You remember that, very important. Okay, so I'll go with the fourth biomacromolecule, very important, nucleic acids. Okay. So what is this nucleic acid? See, I have written acid. That means a chemical which must be acid here. Okay, so there are only two types of nucleic acids in the entire world. What are they? Simple. DNA, which is our genetic material, and RNA, which is also a genetic material in some viruses. Okay, DNA, RNA both are acids. They are nucleic acids. Okay, if you look into the structure of DNA, so let me just give you a simple structure of DNA. DNA looks like a ladder which is double stranded, whereas RNA normally single stranded. Sometimes they also make a double stranded. Okay, so not necessary here. Whatever it is, what is common here is this long line and some line which is actually projecting towards the center, correct? So which is perpendicular to this line. Okay, this is actually sugar phosphate backbone and this is the nitrogenous bases. That's it. Even in the RNA also same. This is actually long strand which is containing sugar as well as phosphate, and a line which is perpendicular, that is nothing but nitrogenous bases. That's it. Whatever it is, I'm just going to take one small structure which looks like this. So this is actually makes long chain of DNA as well as long chain of RNA. So this is common to both the DNA and RNA. What is this? This is called as nucleotide. Yes. What is this nucleotide? It is the structural as well as functional unit of DNA as well as RNA. Many nucleotides. If you take one nucleotide here, I'm going to draw one small T-like structure. This is one nucleotide which containing three components. Can you please name the three components? Nitrogenous base, the first component. Second component is pentose sugar, and the third component is phosphoric acid or phosphate group. See, phosphoric acid gives acid to this chemical DNA or RNA. So deoxyribonucleic acid, ribonucleic acid, acid because of phosphoric acid present in them. Okay, so nucleotide is common to both DNA as well as RNA. So let us see nucleotide components. Mainly nucleotide, if you take this structure, it is made up of three components. What are they? The first important component is nitrogenous bases. That is a chemical which is containing nitrogen. It is normally a heterocyclic ring. I had already discussed in the classroom. Okay, I am not going to discuss in deep now. Anyhow, in second PUC, you are going to study the same concept in detail. Okay, so in nitrogenous base, there are again two types. Okay, so what is that two types? The first one is called as the purines. The second type of nitrogenous base is called as pyrimidines. Purines and pyrimidines are the two types. Okay, so what is this purines? Again, purine is made up of heterocyclic nitrogenous rings. Actually, it looks like this. So two different types of cycles are there. The benzene rings are there. Got it. So that is purine. What are the different types of purines? Okay, present in nucleic acid. That is adenine, very common one, and the second one is the guanine. Yes, this is very common. Okay, pyrimidines, which is not heterocyclic ring. Normally one ring will be there. So pyrimidine is there are again three types. Let me just name it. Very common: cytosine and thymine. Adenine, guanine, cytosine, thymine, that is purine and pyrimidines, normally seen in DNA. In RNA, instead of thymine, we have one more pyrimidine, that is uracil. So adenine, guanine, cytosine, uracil is seen in RNA. Adenine, guanine, cytosine, thymine is seen in DNA. Simple. Okay, so that's it. We'll go to the next important type, that is, let me just write. I forgot to mention one thing. You get one question for your competitive from this part, that is, name the nitrogenous base which is present in DNA but absent in RNA. Thymine is actually present in DNA. Okay, in RNA you don't see thymine, instead of that you see uracil. The question could be asked vice versa. Okay, you should be careful here. Okay, so we go to the next part, that is the second part of the nucleotide, that is pentose sugar. So what is this pentose sugar? The sugar which looks like this. Hope you can see this. This is actually the pentose sugar. Got it? Okay. So in the case of DNA, you don't see oxygen here in the second carbon atom. Okay. In the case of RNA, you see oxygen here. You will learn in the second PUC that because of the presence of oxygen in RNA, which makes highly reactive chemical, so that RNA cannot be a stable compound. Got it? Whereas DNA is very stable, it will not undergo mutation rapidly like RNA. Got it? Because of the absence of oxygen in DNA. Okay, so this is pentose sugar now. Fine. Let me just write this point. In DNA, what type of pentose sugar you see? Where oxygen is absent, so we call it as deoxyribose sugar. Where in DNA? In DNA you see a pentose sugar that is called as deoxyribose sugar. In RNA, oxygen is present, so simple, it is nothing but simply we write ribose sugar. That's it. It is having oxygen, so we don't write deoxy. Got it? So this is actually in RNA. So this is actually the pentose sugar in nucleotide. Okay, we go to the next further third type, that is actually a very important one, that is phosphoric acid. Phosphoric acid, or in a simple word I can write this as phosphate group. Okay, phosphoric acid, phosphate group. How it looks? Chemically it looks like this, O, 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 and one oxygen is carrying negative charge. Now the DNA is particularly negatively charged. Why DNA is negatively charged? Because of the presence of phosphoric acid. And DNA or nucleic acid, it is called as acid. Why? Because of the phosphoric acid. Got it? Okay, so DNA is negatively charged because one of the oxygen molecule is here in the phosphoric acid is negatively charged. Done, which gives negative charge to the DNA. So this is the basic. Now let me just continue further. For example, let me just write a word here. Okay, simple. That word is nucleoside. What is this nucleoside in DNA or RNA? Very simple. Okay, let me just write. When any nitrogenous base, it could be adenine or it could be guanine or it could be cytosine or thymine or uracil, it doesn't matter actually. Okay, whether it is in DNA or RNA, that is also doesn't matter. Okay, when a nitrogenous base is linked to a pentose sugar, this sugar, when a nitrogenous base is linked to a pentose sugar, then the structure is called as nucleoside. For example, here, let me just write here. When adenine, okay, let us assume that adenine is here in this place, in the nitrogenous base. Okay, when it is linked to a pentose sugar, the pentose sugar is now common to any type of nitrogenous base. Okay, pentose sugar, and this structure is now called as adenosine. Simple. So adenosine is nothing but a nucleoside. Okay, so we can just go on making one more type, that is another nucleoside. If guanine is present in the nitrogenous base place, so with pentose sugar, and you call it as guanosine. Simple. Yeah. Okay, or you can go with the cytosine with another pentose sugar. You cannot call it as cytosine like this. You have to call it as cytidine. You call it as cytidine. Cytidine is the nucleoside. Okay, so next one is we'll go with another one, that is thymine with another pentose sugar. The pentose sugar is common actually. Okay, different pentose sugar. Okay, thymine, you call it as thymidine. Simple. The next one, the fifth one, that is uracil. In the case of RNA, okay, with the pentose sugar, you just call it as uridine. I hope that you are copying all this stuff. I'm not providing special notes actually. Okay, so simple. So these are the different nucleosides. Okay, now I just want to tell you that what type of the bond is present in the nucleoside. There is a linkage, a bond is there. What kind of bond? See, there is a carbohydrate that is in the form of pentose sugar with a nitrogenous base. This link is actually called as N-glycosyl acidic linkage or even bond also, N-glycosidic bond. Okay, so please note this point, very important for your competitive part. What is the bond present between a nitrogenous base and a pentose sugar? Or the question could be directly, that is name the bond in nucleoside, N-glycosidic linkage. Here all whatever you see here, the nucleosides, there is a linkage called as N-glycosidic linkage. Got it? Okay, so we go to the next part. When a nucleoside, that is two parts, nitrogenous base and a pentose sugar, linkages with the phosphoric acid or a phosphate group, what you call now? You call it as nucleotide. Simple now. So finally we got a structure which looks like this. This one, the line which is perpendicular to this vertical line, this is nitrogenous base. This one is a pentose sugar, and this one is nothing but the phosphoric acid. So three components are there, and that component is called as nucleotide. Here a nucleoside links with a phosphate group, and the entire structure is called as nucleotide. Okay, so this is the basic unit of polynucleotide chain, the entire chain. Okay, so here I just want to tell you one more thing here. Any type of nucleoside, you take this one, that is adenosine. Okay, let me just write here. Okay, we have one nucleoside here, that is adenosine. When it links to a phosphate group or phosphoric acid, let me just write this, phosphate group. Okay, so what you call that? You call it as adenolic acid. See, now it has become acid, adenolic acid. Okay, so we go to the next one, that is when another nucleoside, that is guanosine, links to phosphate group, and what you call it as guanilic acid. Okay, next one, that is when another nucleoside, that is cytidine, links to a phosphate group to make a nucleotide, and this nucleotide is called as cytidilic acid. The next one, we go with the thymidine, another nucleoside, that is where nitrogenous base is thymine. Okay, when it binds to pentose sugar, it makes thymidine. When thymidine binds to or links to the phosphate group, and you call it as thymidilic acid. Done, easy. The next one, the last one, in the case of RNA, when uridine binds to the phosphate group, what you call it as uridylic acid. Done, easy. So this is what actually entirely called as nucleotide. Now this is nucleotide. Please write down everything, all components, whatever I have discussed is very important. Now I have to tell you the linkage. See, when a nucleoside binds to a phosphate group to form nucleotide, and there is another linkage, the second linkage, that linkage is actually called as phosphodiester bonds. In a simple word, you can also write like ester bonds, but I'll go specifically, that is phosphodiester bonds. Okay, we'll highlight this. So the bond is present between a nucleoside and a phosphate group is phosphodiester bond. So you have to be thorough with N-glycosidic linkage and phosphodiester bonds. Very important. No confusion here. Very simple, right? Nucleoside, N-glycosidic linkage. In the nucleotide, phosphodiester bonds. For example, let me just write here. So this is one nucleotide. Okay, this is another nucleotide. This is another nucleotide, and now they are linked now. Okay, this is the linkage. The linkage is mainly phosphodiester linkage. Here you see phosphodiester linkage. Between the two nucleotides, there is a linkage called as phosphodiester linkage. Done. Okay, so this is very simple now, and many nucleotides ultimately make a long chain, okay, which makes single strand of the nucleic acid. It could be DNA or RNA. So we just name this side, one end of the strand is named as 5 prime. Okay, this is the other end of the strand, we call it as 3 prime. That's it. And I'm going to draw one more line which makes DNA here. Now this is double strand, and this strand cannot be called as 5 prime. 3 prime. It should be called as 5 prime, and this is 3 prime. Simple, easy. In the case of RNA, you see a single strand like this. So this is what you see, RNA 5 prime and 3 prime. Done. So this is what the basic information regarding the nucleic acid, but in second year PUC you're going to study everything. Got it? Okay, it's done. Now I'm going to discuss the double helix model of DNA. Very important. Now let me just explain that double helix model of DNA. Who proposed that? Watson and Crick in 1953. Double helix model of DNA. Very important. Okay, so first we write the scientist who proposed this model. The name of the scientist, Watson and Crick. James Watson, Francis Crick. They actually proposed this double helix model when in 1953, and even they won Nobel Prize also. Okay, in detail you will learn in second year PUC Nobel Prize. Let me write here. Okay, so before I explain the structure, let me just give you the diagrammatic representation of the double helix model, which looks like this. You also draw this. See, okay, which looks like this. So this is what actually the double helix model. And what is this now? This is now nothing but single strand. Okay, let me write 5 prime one end. Okay, and this one is the 3 prime end, and this one is 5 prime and this one is 3 prime. So it is understood that one strand is running in this direction, the other strand is running in opposite direction. So both the strands in the DNA are anti-parallel. Simple. Okay, so anyhow, I'll come to that point. Now let me just write another important point of double helix model. Here, how many strands are there? 2 polynucleotide chains are there, correct? Chains are present and they run anti-parallelly, opposite. Okay, I think, yeah, anti-parallel, fine, no issues. Okay, and one more thing is these two strands are actually linked by what? Nitrogenous bases. That's it. These nitrogenous bases, they join these two strands. Okay, like this. So it goes on like that. It looks like right-handedly coiled staircase, correct? So it looks like a staircase, but which is right-handedly coiled staircase. Simple. I think I made a mistake. Staircase. Case. Okay, fine. The next one, let me just go with the next point, that is, what is the backbone of this entire DNA? The backbone. This is actually the backbone, which is nothing but polynucleotide chain. The backbone is nothing but sugar and phosphate, not the nitrogenous bases. Sugar and phosphate, which makes the backbone of DNA. The backbone. Got it? So sugar and phosphate, which makes a backbone. Very they are very strong. Actually, they gives rigidity to the DNA. Okay, it prevents collapsing of the DNA. This is it. Okay, now one more thing is the nitrogenous base. You could see here there are 2 nitrogenous bases, a pair of nitrogenous bases, okay, present in one particular structure, which is actually perpendicular to this backbone, correct? Okay, so this is the nitrogenous base which is perpendicular to the backbone, right on this point, which is perpendicular, correct? Okay, fine. Now if you take the nitrogenous base, that is the purine, for example adenine always links to thymine, or vice versa. Thymine always bonds with or pairs with adenine with the help of 2 hydrogen bonds. See here, it is not equal symbol, it is 2 hydrogen bonds. Now you get one more question. What is the type of bond which is present between 2 nitrogenous bases? That is between purine and this is pyrimidine, correct? So let me write in the bracket, this is purine and this is pyrimidine. Okay, purine cannot bind to another purine. Adenine cannot bind to guanine. Got it? It always should bind to the pyrimidine. Okay, adenine, thymine with the help of 2 hydrogen bonds. Okay, for example, here if I write adenine and here I cannot write guanine, I should write only thymine. So there is two hydrogen bonds. Okay, another thing, the guanine. So what is guanine here? The guanine is purine, correct? Okay, it's a purine which always binds to cytosine. That's it. Okay, so guanine always binds to cytosine in DNA with the help of three hydrogen bonds. So let me write here. So you may get a question, if the DNA, in the DNA, if there are more number of guanine is present, then it is a fragile DNA or a very strong DNA. It's very strong DNA. Why? Because guanine always binds to cytosine. If more guanines are there, then obviously cytosine must be there, and the bond is also more. Actually, three hydrogen bonds are there. More the hydrogen bonds, the stability of the DNA increases. It's very difficult to break the DNA. Got it? If you have more number of adenine, then the DNA is weaker. Simple, because only two hydrogen bonds are there. That's very simple. Okay. Now, if we just take, let us assume that this is the first point of DNA. So simple for our understanding. The first point, the starting point of the DNA, and this is the end of the DNA. So from here to here, this is actually called as one turn of DNA. Correct? Correct. Yes, this is the one turn. Okay. If you count it, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. How many nitrogenous base pairs are there here? 10 are there. So let me write, in one turn of DNA, how many nitrogenous bases are there? 10 nitrogenous base pairs are there. Correct? This is very, very important. Okay. Between the two nitrogenous base pairs, so this is one base pair and this is the other base pair. Between the two, there is a distance of 3.4 Armstrong units. So let me write here. Between two nitrogenous base pairs, what is the distance? 3.4 Armstrong unit. So this is the thing. So can you please tell me now, what is the total distance from the starting point of the DNA to the end of the DNA in a one turn? So what is the distance of one turn of DNA? So from here to here, very simple. 3.4 is the distance between the two nitrogenous bases. Okay, so 3.4. How many nitrogenous base pairs are there? We have 10 base pairs. So now you get 34 Armstrong units. Very simple. So distance of one turn of DNA is 3.4 into 10 base pair, that is equal to 34 Armstrong units. Simple. So that is the distance. Done. Hope you got this point actually. So this is all about the double helix model of DNA. I'm going to talk about the types of DNA. There are actually 4 types of DNA have been discovered. I'm not going to discuss here in the first PUC. In second PUC, I'm going to talk about it. Whatever I explained is actually belongs to a type of DNA which is called as bDNA. So this is bDNA structure. Okay, even aDNA is there, cDNA is there, zDNA is there. zDNA is the bigger DNA. Okay, so this is what actually present in all living organisms. Okay, so you will understand it later. So this is all about the double helix model of DNA. So we have discussed nucleic acids, and I'm sure that you have no doubts and the entire concepts are clear. Okay, and this one also. I hope that you have taken the notes also. Okay, maintain the notes. Okay, done. So now we will discuss another concept. Okay, so that is metabolism and metabolites. Okay, so we'll continue. Now we will discuss another important concept, that is metabolism. Here you are a bit familiar with this word, that is metabolism. You have recently studied respiration in plants as well as the living world chapter. In that, the metabolism term was there. What is metabolism? Very simple. It is the sum total of chemical reaction. Correct? Total of chemical reaction. Because your body and even my body is made up of chemicals. Okay, millions of chemicals. There is a continuous reaction occurring, and ultimately which makes a state in the body, and ultimately we call it as life, living organism. That is the sum total of chemical reaction occurring in the living organism. Okay, so metabolism. What are the types of metabolism? We know that the one is actually called as anabolism. I told you how to remember anabolism. Anabolism is AC. Catabolism is CD. C, catabolism. D means destruction. Anabolism. A means anabolism. C is constructive. anabolism is always a constructive type of metabolism. That means it builds the chemicals. Got it? From tiny chemicals, a big chemical is formed, that is anabolism. Okay, I'll write the second type, that is catabolism. Glycolysis is a good example for catabolism, where from a complex material, chemical, which ultimately broken to form simple chemical, that is glucose, ultimately forms pyruvic acid. That is catabolism. Glycolysis is catabolism, which is called as destructive process. Destructive. Simple. Okay, so I'm not going to talk about anabolism and catabolism here. You know about it. Whatever it is. So for example, let me write A plus B. These are the two types of chemical which makes another chemical. Okay, or another type of chemical reaction could be AB can form A plus B. So these are the two types of chemical reaction occurring here. Correct? Okay. Anabolism, this one. Catabolism is this one. Okay. Whatever it is, what is required? You have studied glycolysis, in that you have studied the 10 steps. Each step is carried by a specialized or specific enzymes. Correct? Okay. So this is what I want to tell you here. Without enzymes, the chemical reaction cannot take place. Even if it takes place, it takes a lot of time. If you just add enzyme, the chemical reaction takes place within a fraction of a second. This is it, because enzymes got a turnover number. Okay, so we'll talk about enzymes later. Fine. Okay, so here just want to write the metabolites. What is this metabolite? Okay, so let me write here metabolites. What is this metabolites? All we have discussed in this chapter: amino acids, proteins, polysaccharides, okay, lipids, even nucleic acids, all are metabolites. Okay, so in a simple word I can write these are organic compounds. Organic compounds present in a living organism. So they play an important role in metabolism. Okay, some are required for metabolism, to run the metabolism. Some are the product of metabolism. That's it. That is nothing but metabolites. Okay, so now we can classify the metabolites into two types. This is very important for your this exam NEET. Okay, so please note this point. There are two types of metabolites. The first type is called as the primary. Okay, primary metabolite. Okay, here I'll write the second type of metabolite is called as secondary metabolite. You get a lot of questions from primary as well as secondary metabolite, particularly the example. Okay, so we just put a box here, primary metabolite, and this one is secondary metabolite. Done. Okay, so let me just list out the difference. Okay, so we just put a line here so that we can classify them. Okay, so a bit of disturbance is there. Okay, fine. And now what is this primary metabolite? The first one, these are the chemicals, organic compounds play an important role in metabolism. They are required for metabolism. Okay, this one, the secondary metabolites, they are exactly opposite. They doesn't play, I think you can continue, any important role in metabolism. So this is what the first point, you should know the difference. Okay, primary metabolite. Okay, second important difference is they are required to run any type of chemical reaction, that is nothing but metabolism. Okay, required to run metabolism. Here, no, not required to run the metabolism. Got it? Okay, and this one, they have very specific role. So I'll write here specific role in living organism. They have specific role. For example, you take GLUT4, a type of metabolite which is primary metabolite, which acts as a receptor. I had already discussed, okay, in the protein concept, I guess. Yes. Okay, so the GLUT4 actually receives glucose so that the cell can take up the glucose. So that is actually a primary metabolite. They have a specific role. You take hormones, they are primary metabolites. You take enzymes, they are primary metabolites. Okay, they have roles. Okay, in this case, no, that doesn't have a specific role. Okay, Examples. So let me just write the fourth difference, that is our examples for primary metabolite. Okay, any type of proteins, hormones, enzymes, okay, or receptors, that is nothing but protein here. And carbohydrates, any type of carbohydrates present in the body. Okay, even the fats, they are nothing but fatty acids and glycerol. Okay, and even the amino acid. Okay, amino acids actually make up the proteins, but I'm talking about individual amino acids. These are all primary metabolites. Done. You are not forgetting. Very important. Please note all the point. Example for secondary metabolite. There are so many examples. I'm going to give you the list of examples. Okay. So let me write alkaloids. Alkaloids. Okay. Okay. Fine. Alkaloids. Then one more example is flavonoids. Okay. Even rubber. I think you know rubber, okay, which is helpful in making tires and other things. Okay, even certain oils, that is essential oils, sunflower oil, yes, okay, castor oil. So this is essential oil, plant product, okay, which is secondary metabolite. Even antibiotics, certain fungi have the ability to release chemicals, which is not helpful helpful for the fungus, but it is helpful for human beings, so that we can take this antibiotic, a chemical produced from the fungi, to kill a bacteria. Okay, penicillin, for example. Yes, antibiotics. Even the pigments. There are so many pigments, right? Okay, pigments like in beetroot, you see this type of chemical called as anthocyanin. There is no specific role in beetroot, that is by anthocyanin. Okay, so it is secondary metabolite. Okay, or even scents, perfume, scents, gums. Okay, even the spices. Please note down all this point. Actually, these are all examples for secondary metabolite. So you should know the difference between primary and secondary metabolite. Hope you understood this. Very simple actually. Primary metabolite is nothing but a chemical which is very important for the body. Secondary metabolites are not at all important for that living organism. It could be useful for human beings. We can extract that secondary metabolite, the chemical, from the plant body, or maybe from the bacteria, or maybe from the fungus. We can use it. Okay, so for medicinal purpose, or maybe for commercial purpose. For example, you could see here rubber, commercial purpose. Okay, or antibiotics for medical purpose. That's it. Okay, now I'm going to give you the list of secondary metabolite. Okay, this is very important, so let me just close this. That means end of the concept. Now there is a bit of work for you. You need to mug up this. I'm going to give you the list of secondary metabolite. It could be asked in your competitive secondary metabolites. Some list. There are about 8 to 9 list. Okay, if I remember, I think I can list out. Okay, the first one, let me just write the pigments. Example, specific example for these pigments: carotenoids. Okay, even you can also write anthocyanin. Mug up this: anthocyanin, carotenoids, pigments. Okay, so second type is the toxin, certain toxic materials. Okay, that is abrin, ABRIN, then ricin. Okay, abrin, ricin are the type of toxins, plant toxins. Certain drugs, there are so many drugs actually. Okay, so that is curcumin, vinblastin, VINBLASTIN. Okay, the next one, polymeric substances. That is nothing but rubber, even the gums, even cellulose. Okay, cellulose. Okay, fine. Next one is, let me write, terpenoids. This is very important. You get a question, terpenoids. Okay, so we put a box here. Another type of secondary metabolite, terpenoid. You can write monoterpenes or diterpenes. Some examples. Okay, the sixth one, I hope it is visible to you now. The sixth one is alkaloids. Okay, it's morphine. Morphine is very important for the production of brown sugar. In second piece, you are going to study about it. Okay, or there is one more actually, that is codeine. Not required, but remember morphine, which is a type of alkaloid, a secondary metabolite. The seventh one, I think, lectins. lectins. This is important. Lectin example, concavalin, concavalin A. Concavalin A is a very good example for lectins, another type of secondary metabolite. And I think the last one we have essential oil, essential oils. Okay, so for that you can write lemon grass oil. Done. So this is what you need to write down this and mug up. Okay, make write like this and just stick it to any of your wall. Okay, recall it every day because it's very important. Okay, even for me also it's very difficult. Okay, I prepared and I had written everything, but for you, like you need to prepare for your need, so you need to recall this every day. Okay, I may forget after 2 to 3 days, I may forget because I don't mug up this. Got it? Okay, so this is all about the metabolites, primary, secondary, and some examples for secondary metabolites. Hope you understood. So this is for today. Tomorrow we'll be discussing enzymes, another important, very important concepts. Please learn and make notes. No need to make long notes. Just copy whatever I have written here and just practice. Done. Thank you.